Okay, today we're going to talk about active transport, and active transport is the opposite of passive transport. Active transport is where cells are going to move things in and out of the cell membrane, but this time they're using energy. And the reason they're using energy is because the cell is going to be moving things from a low concentration to a higher concentration. So it's moving against the concentration gradient. It has to pump those or do something to move those materials in that direction. We're going to talk about three types of active transport. That's protein pumps, endocytosis, and exocytosis. And one way to kind of think about the difference between passive and active transport is passive transport that we talked about in an earlier screencast is like going downhill, like rolling a ball downhill. It doesn't require any energy on your part. The ball is going to naturally roll from a high area to a low area, and that's what happens during passive transport. The three types of passive transport we talked about in the previous screencast were diffusion, osmosis, and facilitated diffusion. Active transport, on the other hand, is like pushing or rolling a ball uphill. You're going to have to use some energy because it's going in the opposite of its natural direction. And again, today we're going to talk about endo and exocytosis and protein pumps because all three of those processes move things from a low concentration and build it up to a high concentration. So our first one is the protein pumps, the cell membrane pumps. This is when here in the cell membrane there are pumps that will move proteins and push them in and out of the cell. And unlike the channel proteins for facilitated diffusion, these have to have energy. The energy allows the protein pump to change its shape, and when it changes its shape, it pushes the material in or out of the cell. And each pump is made to fit a particular particle, a particular molecule. So not just anything can go through that particular pump. It has to be a certain shape. It has to be that particular type of molecule. Once the particle is released, the pump's going to return to its original shape. If it's going to change shape and move material in or out, again, energy is going to have to be used by the cell. Our second type of active transport is endocytosis, and endocytosis is taking in large molecules from the environment. And our prefix endo again means in. It's how materials are going to enter the cell. So you can see here there's material, and what happens is the membrane kind of folds around it, the cell membrane, and it packages it up into either a vesicle or a vacuole and moves it through the cell. And it does this for larger particles, like maybe bacteria can be destroyed this way, food can be taken in this way, oil droplets, cholesterol, those types of things. The opposite of endocytosis is exocytosis, and exocytosis is where the material is being pushed out of the cell. It's exiting. Again, exo means exit. And this is how cells can move big materials out of the cell. It's also how they will get rid of wastes. And the exact opposite again happens. The vesicle containing the material comes over, merges with the cell, opens up, and releases that material into the environment. So that's our third type of active transport. Here you can see an actual microscopic image. Here's the vesicle after it's merged with the cell membrane, and it's releasing its contents into the environment. Exocytosis and endocytosis are important for all cells, but they're really important for unicellular organisms like this amoeba. So this is how the amoeba gets its food. It surrounds it through endocytosis, brings it in, digests it, and then the wastes are gotten rid of through exocytosis.